The story of genetics begins with an Austrian monk. His name was Gregor John Mendel, a monk who turned into a scientist and laid the foundation of modern genetics. He worked on pea plant, Pisum sativum, and he died as an obscure scientist. He was not known in his lifetime. But his laws were put forth by his own name later in 1901 by three scientists. One of them was Hugo de Reis, who gave the theory of mutations also. Then the second one was Karl Corens, and one more name who rediscovered Mendel's laws. He was von Schermark. History of genetics can be divided into three parts. It begins with the Mendel's laws and the representation or publishing of his laws after his death, almost 17 years after his death. Then the second step begins here, and that is the study of chromosomes. The study of chromosomes, this period, it continues in the first 2 decades of 20th century. It includes the work of Sutton and Boveri. It includes the work of the famous fly lab of Thomas Hunt Morgan. And one more name comes here, and he is Sturtevant. He was student of Sturtevant, was student of Thomas Hunt Morgan, his colleague, and he gave the concept of gene locus. Genes are located on chromosomes on specific loci, specific places, and this is one important discovery in the history of genetics. We can give the gene's address. For example, a gene is present on 3rd chromosome or 5th chromosome, its shorter arm or its longer arm, and the specific bands and interbands after that. So this is interesting, the second part, when chromosomes were studied comprehensively. A science of cytogenetics developed in this period. And the third phase is DNA molecule, the discovery of molecular structure of DNA by James Watson, Francis Crick, and Maurice Wilkins, that was based upon study of Rosalind Franklin. And this period also includes the duplication of DNA, then the concept of genetic code, and that comes up to the Human Genome Project. So, this is the third segment of genetics. This now diverges into various different fields like the applied genetics, means DNA recombinant technology, etc., etc. Mendel was born in a poor peasant's family. He lost his parents in early age. And then he had to go to a monastery that was closer to his native place. And he lived there further. He took religious education and other schooling was done there. Mendel's formal education took place in a school attached with that monastery. And then he decided to continue there as teacher. At early age of 25, he became a monk. He was given the oath of celibacy and poverty according to the theological system of Christianity. He became Gregor then. So, there is an interesting dot of this Western theology that connects even with Eastern one. See, in all religious notions, we find that poverty is an important aspect that is associated with the religious preachers. The people who preach the religion, they are so simple. They live with least possible belongings. In India, these people are called Parivrajak. They are described by these Sanskrit lines, that is, Bhuprasthachari vidhilabdha toshaha. They wander without any belongings, but they are like kings. Swami Vivekananda used to sing this song when he became a sannyasi. Vedanta vakyeshu sadaramanta kaupinavanta khalu bhagyavanta. A monk without any belongings, but he is the king of this entire universe. This is the Eastern notion about monks in India. Why? Even in West we see that there is given an oath of celibacy and poverty. And here this poverty means to depend upon the least possible things and do the work of that religion or work for that particular religion. So Mendel then began teaching in the school that was associated with that monastery. And he thought that he should take formal education and decided to go to Vienna for his further school certificate course. Here begins the transformation of this young monk into a scientist who laid the foundation of the greater picture, the larger picture of genetics that we see today. He gave 3 laws of inheritance. In fact, he statistically represented his theory. In any scientific discovery, the results have to be put in a very tangible manner. And Mendel was successful in that, but he was unfortunate. Lot of the times, a researcher in science, he needs some endorsement from other people from scientific community, but that did not happen. He communicated scientists like Nageli in that period, and he told about his observations in Pisum sativum. But here still he is unfortunate. Nageli denied to accept the scholarship of his work, the merit of his work. Well, Mendel continued to work, and he died in 1884. He was unknown to the scientific community for a greater period of time. Till 1901, his papers were republished, and of course by Mendel's name itself, by these three scientists, Hugo de Ries, von Schermark, and Karl Korenz. Many scientists independently tried to count the number of chromosomes. Some of them were fortunate because those organisms where they tried to count the number of chromosomes, they had few pairs, hardly some 2 pairs or 3 pairs or some 5 pairs of chromosomes. So independently, different scientists, they tried to collect this data. They tried to count the number of chromosomes in different organisms. So in almost 1923, after 20 years of Saturn and Boveri's theory, human cells, they were taken for study. The count of chromosomes that was in human cells, human somatic cells, that was perhaps searched by these scientists in that period. And there established a notion that human cells have 48 chromosomes, 24 pairs, and this continued for almost 20 years later. 22 years, you can say. In 1956, Tijo and Levon, they gave the correct count of chromosomes in human beings. See, this is after the discovery of molecular structure of DNA, almost 3 years after that. Why I am emphasizing this point? The reason is, look at this span of some 20-25 years. This is the period in which the microscopy evolved. It became more and more ultra microscopic studies, those started in this period. In 1931, Knoll and Ruska, they came out with electron microscopy. In 1938, they developed the methods like X-ray diffraction studies in England. Ostbury in 1937 came out with this X-ray diffraction studies, and the scientists could take pictures of ultra microscopic structures, and that was the basis of Watson and Crick's discovery or research. Maurice Wilkins, he shared a Nobel Prize with these two people. But the foundation of this work are those images by X-ray diffraction studies. Those were taken by Rosalind Franklin. In the science magazine Nature, in the April issue, the molecular structure of DNA was published by James Watson and Francis Crick. Look, the microscopy was evolved to such an extent. This was the period in which the viruses were crystallized first these 20-25 years. An interesting aspect of this thing is study of these viruses that in fact began in India. A British scientist, Hankin, he found that the water in Ganga and Jamuna rivers, this water is having something unknown that kills the vibrios, the bacteria, those cause cholera. Then Bijering, he suggested the concept of a filter past microbe that cannot be seen via any microscopy, through any microscopy. So in this period, in between these two, these results from India, they were published by Hankin in 1896. So that was a period. You can correlate that even with the germ theory of diseases, because 1896 is almost 22 years after the Koch's theory. Robert Koch, he gave his theory of what we call as germ theory of diseases. Chromosomes appear only during cell division. Otherwise, DNA is scattered with the proteins, what we call as histones, in eukaryotic cells. The chromatin is made up of DNA and histone proteins. This structure is like a beaded string that further coils and supercoils, and then at last it produces the most condensed form called chromosomes. This condensation of DNA into a chromosome, you can compare that with a reel where thread is coiled upon a core. So similarly, that reel represents chromosome while that thread represents DNA. Our cells, each cell, every cell, it has a very long DNA. DNA in our single cell is longer than our height. It is almost 1.5 times or little more longer than how tall we are. The length of DNA is enormous, but its diameter is less. So as it condenses, it gets coiled, supercoiled using the histones. It condenses into simple rod-like structures, what we call as chromosomes. These chromosomes appear during the cell division. See, our cells, they work in two phases. The first phase is the phase in which the DNA is transcriptionally active, means it produces messenger RNA and then proteins in the cells. So for that, the DNA should make a template and it must not be condensed. But during the cell division, just before the cell division begins, this DNA, it condenses, it shrinks into a rod-like structure, what we call as chromosomes. Each species has a fixed count of chromosomes. We humans, we have 23 pairs of chromosomes. So, how do we understand them? Look, there are 46 DNA molecules, 23 pairs of DNA molecules in a cell. And each piece of these DNA molecules, when it condenses, it makes a chromosome. So, technically, the longest chromosome is the first chromosome. Then, shorter than that first chromosome is the second chromosome, then third chromosome, and so on and so forth. So, chromosomes are numbered in the decreasing order of their length. This method of making a chart of chromosomes using the decreasing order of their length is known as karyotyping. That chart is called a karyotype, and this method is called karyotyping. See, when we are observing chromosomes under microscope, they must be seen distinctively. It becomes difficult to count when the number of chromosomes in that cell is much high. If there is a large number of chromosomes inside the cell, then while fixing them on a slide, it becomes difficult to see them separately. And that is why their count becomes almost impossible, or it becomes incorrect, in fact. So this continued from 1923 to 1956, that human cells have 48 chromosomes, which was wrongly put up in 1923. Even at the time of Watson and Crick, the number of chromosomes was wrongly understood to be 48 and not 46. So what happened in these 3 next years? Look at this. How very simple ideas, they are important in science. They are important in advanced science. The concept of osmosis we learn in our high school days. That is movement of a pure solvent across a semi-permeable membrane from low solute concentration to high solute concentration. This movement that takes place across a biological membrane is known as osmosis. So if we are putting a cell into a hypotonic solution, then what will happen? In hypotonic solution, quantity of solvent is more, means water is more. This water would rush into the cell by the process of osmosis. In animal cells, that would result into bursting out of these cells. These cells would rupture because of entry of water from the surrounding hypotonic medium into the animal cell. But when we see that in plants, the plant cell membrane, it has a cell wall around it. And this rigid cell wall prevents the bursting of that cell. Rather, it becomes turgid due to a pressure, what we call as turgor pressure. But animal cells, if they are put into hypotonic solution, they would rather burst. But what if we keep the amount of solvent very modest and solute more? Then such solutions will have hypotonicity, they will be hypotonic, but yet the quantity of solvent will not rupture that cell. This is something called hypotonic shock. When the cells were given hypotonic shock, the cells swelled, but they did not rupture. That resulted into separation of chromosomes apart from each other. A dividing cell was taken. Its division was arrested at metaphase, and all those chromosomes they got scattered there. See, the amount of DNA that is present in chromosome, uh, that is doubled during cell division. So we see a chromosome with two alike chromatids. So this increases the crowd of chromosomes within the cell. A hypotonic shock, it separates them. Simple osmosis, something that we study in high school. But this helped, this hypotonic shock method, this helped to count these chromosomes, those were scattered. And it was possible to get the correct count of chromosomes in 1956. This was done by Tijo and Levan in 1956. And one more interesting point is associated with this thing. In 1961, scientists came out with the concepts like aneuploidy, means some conditions like Turner syndrome or Down syndrome. Those were caused due to trisomy or monosomy of chromosomes. So this study began due to this 1956 breakthrough, and this 1956 breakthrough was counting the human chromosomes correctly. In science, in advanced science, in research, see how simpler ideas they work. This concept of endosmosis that we learn in our school days, high school days, that actually helped to develop the field of cytogenetics as well as the diseases like the conditions like Klinefelter syndrome or Turner syndrome or Down syndrome. Those were understood properly because of this correct count. 46 chromosomes in human beings. So thank you for listening me so kindly, friends. This is Lakshmi Raman Oza, and you were watching SBTL webcasts. Let's meet again in the next part. Till then, take care. Thank you.